Multi-target approach counters tumor growth in several cancers. Date: January 20, 2025. Summary. Recent research indicates that a multi-target approach can effectively suppress tumor growth across various cancers. A team from Yale University has developed a novel therapeutic strategy aimed at simultaneously targeting multiple pro-tumor activities within the tumor microenvironment. This significant advancement was published in Nature Biotechnology. Background. The tumor microenvironment plays a critical role in cancer progression. The behavior of tissue surrounding a tumor significantly differs from that of distant tissues. Cancer cells can alter their surroundings, inhibiting immune defenses and creating a protective sanctuary for tumor growth. Traditional therapies often focus on single molecular targets within this complex environment, which can lead to limited efficacy. For instance, many immunotherapies benefit only 20 to 30% of patients, highlighting the need for more comprehensive treatment strategies. Research methodology. The Yale research team, led by Professor Sidi Chen, utilized a gene editing tool known as Cas13. Unlike its more prominent counterpart Cas9, which targets DNA, Cas13 specifically targets RNA and has the capability to address multiple genes simultaneously. The team identified several genes responsible for immune suppression and developed a Cas13 system that could effectively silence these genes. The research involved delivering the Cas13 package into the tumor microenvironments of mouse models. This intervention aimed to reactivate immune responses by silencing the genes that suppress them, thus remodeling the tumor microenvironment and enhancing anti-tumor immune responses. Results. The findings demonstrated that this multi-target Cas13 system significantly reduced tumor growth across 4 types of cancers: breast cancer, melanoma, pancreatic cancer, and colon cancer. The study revealed that the approach not only inhibited tumor progression but also reshaped the immune landscape within the tumor microenvironment, leading to a more robust immune response against the cancer. Future directions. While the results are promising, further research is essential to optimize the efficacy and safety of this treatment method. The researchers believe this technology has the potential to serve as both an off-the-shelf treatment for broad application and a customizable option tailored to individual patient needs by modifying the gene targets. The team is committed to advancing this line of research towards clinical trials, aiming to translate these findings into viable treatment options for patients. Community response. Professor Sidi Chen emphasized the importance of this research on social media, highlighting its potential impact on cancer treatment. The study has garnered significant attention and recognition within the scientific community, indicating a shift towards more integrative cancer therapies. Conclusion. Yale University's innovative research on a multi-target approach to cancer therapy marks a pivotal advancement in oncology. By simultaneously targeting multiple aspects of the tumor microenvironment, this strategy could potentially benefit a larger patient population compared to traditional therapies. Although clinical trials are necessary to validate these findings, this research brings renewed hope for more effective cancer treatments in the future. Sources: 1. Yale University News. 2. Professor Sidi Chen's LinkedIn post. 3. eCancer News article. This comprehensive report outlines the groundbreaking work being done at Yale University in the quest to combat cancer more effectively through a multi-target therapeutic approach. The counterintuitive information in the content revolves around the traditional cancer therapies that predominantly focus on single targets within the tumor microenvironment. This approach has shown limited effectiveness, benefiting only 20% to 30% of patients. The complexity of the tumor microenvironment, which can involve multiple pathways and compensatory mechanisms, implies that targeting a single molecule may not yield significant therapeutic benefits for many individuals. In contrast, the recent research from Yale University introduces a multi-target approach using the Cas13 gene editing tool. This method can simultaneously target several genes associated with immune suppression within the tumor microenvironment. By doing so, it offers a potentially more effective strategy for reducing tumor growth across various cancer types, including breast cancer, melanoma, pancreatic cancer, and colon cancer. The counterintuitive aspect here lies in the fact that while traditional therapies focus on singular interventions, the multifaceted nature of cancer requires a broader strategy that addresses multiple factors at once. This shift in understanding emphasizes the necessity for innovative approaches in cancer treatment that can adapt to the individual complexities of each patient's tumor environment. The research suggests that by utilizing a system like Cas13, which can silence multiple immune suppression genes simultaneously, there is potential to enhance the body's immune response against tumors more effectively than with conventional methods. Furthermore, the notion that a more generalized off-the-shelf treatment could be tailored to individual needs by swapping gene targets is another surprising takeaway. This indicates a movement away from personalized medicine exclusively tailored to individual genetic profiles towards a more flexible, broad-spectrum approach that can still be customized according to specific tumor characteristics. Overall, the study highlights a significant paradigm shift in cancer treatment strategies, moving from a narrow focus to a comprehensive multi-target approach that may improve outcomes for a larger proportion of cancer patients. The research from Yale University on a multi-target approach to combat tumor growth in various cancers presents several significant findings that may be overlooked by many readers. Here are some of the critical aspects of the study, along with their significance. 1. Complexity of the tumor microenvironment. The study emphasizes how the tumor microenvironment (TME) is not just a backdrop for tumor growth but an active participant in cancer progression. This complexity often leads to treatment resistance when therapies target single molecules. Most people may not appreciate how multifaceted the TME is, including interactions between cancer cells, surrounding tissues, and immune cells. Understanding this complexity is crucial for developing effective therapies, as it suggests that a more comprehensive approach is needed rather than focusing on isolated targets. 2. Limitations of traditional therapies. The research highlights that conventional therapies often target one specific molecule, which can leave many patients without effective treatment options. Notably, the fact that only 20 to 30% of patients benefit from current immunotherapies is a critical insight. It underscores the urgent need for innovative approaches that can extend benefits to a broader patient population. Many may overlook this statistic, but it serves as a call to action for researchers and clinicians to seek out more inclusive treatment strategies. 3. Use of Cas13 for multi-targeting. The employment of the gene editing tool Cas13 is a pivotal aspect of the research. Unlike its counterpart Cas9, which targets DNA, Cas13 targets RNA, allowing for the simultaneous targeting of multiple genes. This ability to degrade RNA from numerous immune suppression genes is significant because it could potentially overcome the limitations posed by single-target therapies. Readers might not fully grasp the technological advancements represented by Cas13 and how they could revolutionize treatment protocols by offering a robust means to adjust therapeutic targets based on individual tumor profiles. For potential for personalized medicine, the study suggests that the Cas13 system could be tailored for individual patients by swapping out gene targets. This personalization aspect is crucial in cancer therapy, where a one-size-fits-all approach often fails. The ability to customize treatments based on the unique characteristics of a patient's tumor could lead to improved outcomes and a higher success rate. Many may overlook the implications of personalized medicine in this context, which could significantly shift treatment paradigms in oncology. 5. Modeling in mice. Conducting studies in mouse models is a standard practice in biomedical research, yet the results obtained in these models do not always translate to human patients. While the efficacy of this multi-target approach was demonstrated in mice, the significance lies in the potential for these findings to inform clinical trials. However, This transition from animal models to human application is fraught with challenges that the public and even some stakeholders may not recognize. Understanding this gap is essential for setting realistic expectations regarding the timeline and feasibility of new cancer therapies. 6. Implications for future research. The research team's intention to continue optimizing the Cas13 approach for safety and efficacy before moving to clinical trials is a significant step. The potential for this technology to serve as an off-the-shelf treatment is particularly noteworthy, as it could expedite the availability of effective therapies to patients. This notion may be overshadowed in discussions but deserves attention due to its implications for rapid deployment of new treatments in clinical settings. 7. Broader cancer treatment landscape. The findings from Yale contribute to a growing body of literature advocating for multi-target strategies in cancer therapy. This movement represents a shift in how researchers think about treating cancer, moving away from singular-focused therapies towards integrated approaches that consider the interplay of various molecular pathways. Many might not realize that this study is part of a larger trend in oncology that could redefine treatment methodologies in the near future. In conclusion, the Yale University research on a multi-target approach to tumor growth is significant not just for its immediate findings but also for its implications for the future of cancer treatment. Understanding the complexities of the tumor microenvironment. The limitations of traditional therapies and the potential for personalized medicine can shape how we approach cancer therapy. The insights gained from this study pave the way for innovative treatments that could benefit a larger patient population, ultimately enhancing cancer care and improving patient outcomes. 1. Multi-target approach to tumor microenvironment. The research emphasizes the importance of targeting multiple components within the tumor microenvironment rather than focusing on single molecules. This is crucial because the tumor microenvironment is complex with various pro-tumor actions that can evade traditional therapies, which typically benefit only a small percentage of patients, 20 to 30%. By employing a multi-target strategy, the potential for broader efficacy in tumor treatment is significantly enhanced. 2. Use of Cas13 gene editing technology. The study introduces the use of Cas13, a gene editing tool that allows for the simultaneous targeting and degradation of multiple RNA molecules. This innovative approach differs from traditional methods like Cas9, which target DNA. By silencing several immune suppression genes simultaneously, the researchers effectively remodel the tumor microenvironment, reactivating the immune system and allowing for a stronger anti-tumor response. This represents a significant advancement in cancer therapy methodologies. 3. Demonstrated efficacy across multiple cancer types. In preclinical models, the Cas13 system has shown promising results in reducing tumor growth in various cancer types, including breast cancer, melanoma, pancreatic cancer, and colon cancer. This versatility indicates that the multi-target approach could be applicable to a wide range of cancers, potentially benefiting a larger cohort of patients compared to existing treatments. For future directions and tailored treatments, the research holds promise for further clinical translation and development of tailored therapies. The ability to customize the treatment by selecting specific gene targets based on individual patient profiles could lead to more personalized and effective cancer therapies. As the research team continues to advance this technology toward clinical trials, it opens the door for novel treatment paradigms in oncology, reinforcing the potential for significant improvements in patient outcomes. 1. Combination therapy in cancer treatment. One of the most notable historical cases in cancer treatment that mirrors the multi-target approach is the development of combination therapies, particularly in treating HIV/AIDS and cancer. The use of highly active antiretroviral therapy for HIV exemplifies how targeting multiple pathways can lead to better outcomes. Similarly, in oncology, combination chemotherapy regimens have been established since the 1970s, where drugs such as cyclophosphamide methotrexate and 5-fluorouracil are used in tandem to target various aspects of tumor growth and proliferation, thereby improving efficacy and reducing resistance. 2. Immunotherapy advances. The introduction of checkpoint inhibitors such as pembrolizumab and nivolumab represents a significant historical shift in cancer therapy. These drugs, which target multiple immune checkpoints such as PD-1 and CTLA-4, have transformed the landscape of cancer treatment by enhancing the immune system's ability to fight tumors. Their ability to simultaneously block multiple pathways of immune suppression reflects the principles of a multi-target approach similar to the recent findings from Yale University, where Cas13 targets multiple genes in the tumor microenvironment to enhance anti-tumor responses. 3. Targeting genetic pathways. The case of targeted therapies in breast cancer, particularly with the HER2-positive subtype, illustrates the effectiveness of targeting multiple pathways for improved patient outcomes. The development of trastuzumab Herceptin specifically targets the HER2 protein. But research has also led to the use of combination therapies with other agents like pertuzumab, which targets a different site on the HER2 protein, and chemotherapy. This multifaceted approach has significantly improved survival rates for patients with HER2-positive breast cancer, akin to the multi-target strategy proposed by the Yale research team to tackle tumor growth across various cancers. Future outlook: multi-target approach to cancer treatment. The recent advancements in cancer treatment using a multi-target approach, particularly the research led by Yale University, has opened new avenues for addressing tumor growth across various types of cancers. The implications of such research extend beyond immediate clinical applications and point towards a transformative future in oncology. Here are several key predictions for the future based on current trends and innovations. 1. Increased adoption of multi-target therapies. As research continues to demonstrate the efficacy of multi-target approaches like the Cas13 system, we can expect a broader adoption of these therapies in clinical settings. Traditional cancer treatments have often focused on single targets, which can lead to limited success due to the complex nature of tumor microenvironments. With the ability to simultaneously target multiple genes associated with immune suppression and tumor growth, these therapies will likely become standard practice, providing more comprehensive treatment options for patients. 2. Personalized cancer treatments. Advancements in gene editing tools such as Cas13 will facilitate the development of personalized cancer therapies. Future treatments may be tailored to the unique genetic profile of an individual's tumor, allowing for a more precise targeting of the specific immune suppression pathways relevant to that patient. This could significantly enhance treatment effectiveness and reduce instances of adverse effects associated with less targeted therapies. 3. Integration of AI and machine learning. The integration of artificial intelligence and machine learning in oncology will revolutionize the way multi-target therapies are developed and administered. AI algorithms can analyze vast datasets, including genomic information and treatment outcomes, to identify the most effective gene targets for individual patients. This will streamline the customization of treatments and enhance the predictive capabilities of therapeutic responses, ultimately leading to better patient outcomes. 4. Improved clinical trial designs. The future of clinical trials will likely incorporate adaptive designs that allow for real-time adjustments based on patient responses. As multi-target therapies are evaluated in clinical settings, trials may become more flexible, enabling researchers to modify treatment protocols based on interim results. This could accelerate the development and approval of new therapies, ensuring that effective treatments reach patients more rapidly. 5. Expansion beyond current cancer types. While initial studies have shown promise in treating breast cancer, melanoma, pancreatic cancer, and colon cancer, the principles underlying the multi-target approach will likely be applied to a broader range of cancers. Future research will explore how these therapies can be adapted to target the unique microenvironments of other malignancies, potentially leading to breakthroughs in hard-to-treat cancers. 6. Enhanced understanding of tumor microenvironments. As research progresses, there will be a deeper understanding of the interactions within tumor microenvironments. This knowledge will inform the development of more sophisticated therapies that consider not only the tumor cells but also the surrounding stromal cells, immune cells, and extracellular matrix components. A holistic approach will be essential for effectively countering tumor growth and enhancing the body's natural defenses. 7. Regulatory and ethical considerations. With the rapid advancement of gene editing technologies and multi-target therapies, regulatory bodies will face new challenges in ensuring patient safety and ethical standards. Future frameworks will need to address the complexities of personalized medicine, including issues related to equity and access to cutting-edge treatments and the implications of genetic modifications. Ongoing dialogue among scientists, ethicists, and policymakers will be crucial to navigate these challenges. 8. Global health impact. The broader application of effective multi-target therapies has the potential to significantly reduce cancer morbidity and mortality rates globally. As these treatments become more accessible, particularly in low and middle-income countries, the burden of cancer may diminish. Collaborative international efforts will be essential to facilitate the dissemination of these technologies and ensure that all populations benefit from advancements in cancer treatment. 9. Continued research and development. Ongoing research will be vital to optimize the safety and efficacy of multi-target approaches. Studies will likely focus on identifying the most relevant gene targets, understanding the mechanisms by which these therapies modulate immune responses, and determining the long-term effects of such treatments. Collaboration among academic institutions, pharmaceutical companies, and healthcare providers will drive innovation and improve patient care. Conclusion. The future of cancer treatment is poised for significant transformation with the advent of multi-target therapies. The research conducted at Yale University is a pivotal step in this journey, offering hope for more effective treatments that cater to the complexities of individual tumors. As the field evolves, it is crucial to maintain a patient-centered focus, ensuring that advancements in technology translate to tangible benefits for those affected by cancer. The next decade promises to be a period of remarkable progress in the fight against this pervasive disease.